Hello, we welcome you to the Indian Sign Language enabled virtual lab of CSIR Jigyasa. We hope you enjoy and learn with us.
Hello, hello. What is our topic today? We will understand how is blood testing done? How do we figure out who has AB, or O type blood? Let me explain. A phlebotomist is a trained professional who will use a needle to draw blood from your arm or hand at the doctor's office or a clinical laboratory. The typical method for typing blood involves two steps. The first is known as forward typing. The second is known as reverse typing. These are the two medical techniques used for blood testing. The first step is called forward typing. Your blood cells are mixed with antibodies against type A and type B blood. The samples are checked to see whether the blood cells are sticking together or not. When blood testing is done, your blood is put into a test tube, and when it is mixed with a particular kind of antibodies, and if your cells react to it, it means your blood cells are working with that antibody and they're sticking together. Another example is if your blood cells stick together when mixed with antibodies against type B blood, you have type B blood. The second step is called reverse or back typing. In this kind of testing, the liquid part of your blood without the RBCs or the red blood cells, which is known as serum, is mixed with blood cells that are known to be type A or type B. People with type A blood will have antibodies against type B in their serum. And those with type B blood will naturally have antibodies against type A blood in their serum. Type O blood contains both anti-A and anti-B antibodies. If the agglutination process occurs when your serum is mixed with type B blood cells, you have type A blood. If the same occurs with type A blood cells, you have type B blood. Type O blood, as we know, contains both anti-A and anti-B antibodies. 3 monoclonal antibodies are kept in an icebox, that is anti-A, anti-B, and the monoclonal D. Let us understand the procedure. First, we swab the finger with an alcohol swab and take a blood sample using a lancet. We take 3 separate drops of blood on a glass slide, place a cotton ball on the finger to stop the blood flow, pour 1 drop each of anti-A, anti-B and monoclonal D on each drop of blood respectively. We mix the monoclonal antibodies with the blood sample using a toothpick. When RBCs carrying one or both antigens are exposed to the corresponding antibodies, they clump together. For example, in this slide, agglutination occurred in the first and the third drops, which corresponds to antigen A and Rh factor. That means the blood group is A positive. We can identify the blood group by noticing the agglutination on the corresponding drops.